This lecture is being brought to you in part by the generous gifts of these sponsors.
So I'm used to screaming into a microphone, not having one just kind of wrapped around my face. Thank you very much for the introduction and taking the time this evening to come see me present some of the work that we've done, as well as the cool stuff that others have done. So thanks again for the introduction, and thank you for taking the time to come listen to me talk. Hopefully it will be somewhat informative and not boring. So if you haven't been to Pensacola recently, this is our new research complex. This was the primary thing that led me to come to IHMC, to work in this brand new, beautiful biomedical research complex that we call the Health Span Resilience Performance Research Complex, HRPRC. And I think I'm the first person to get to announce this. On Monday, we finally got our first big grant since this opened, our first NIH grant of $5 million, 5-year project where we get to study how people age 60 and above respond to exercise. So, well, while there's nothing I would love more than to take full credit for that, this was led by Dr. Marcus Baumann, who directs our HRPRC, and in collaboration with OMRF, which is the Oklahoma Medical Research Foundation in Oklahoma City. So the goal is to use this brand new, beautiful building where my office is not working. So my office is on the second floor. But the first floor is basically meant to test how humans respond to stress. In this case, the stress is going to be exercise, and the population will be 200 aged people that will come to the gym to work out for 22 weeks. So this is the kind of stuff that we're doing. Unfortunately, don't get to present anything about it, but this is kind of the future of where research is going and why I joined IHMC. So most of my background has been spinal cord injury. So we're going to talk a little bit about traumatic spinal cord injury, then towards the end get into Parkinson's disease. And I'll kind of explain how does a Spanish major that likes punk music somehow turn into a scientist from animal work to human work to Parkinson's disease. So unfortunately, you have to hear a little bit about my backstory, and then we'll get into some science and some cool data. So I do have a few disclosures. One is through a VA project, we did patent this molecule to work with traumatic injuries of the nervous system. No data about this will be presented, but I think it's fair that I should disclose it. And exercise is awesome. So if you leave this talk going, this speaker is an idiot, I wasted my time, but I should probably exercise tonight, I think that that's a good thing to do. So this is a disclosure that anybody that's in the exercise field does. No drug is better than exercise. Nothing is better than exercise. Everybody should be exercising. That's enough soapbox. But exercise is awesome. And then another thing to kind of spice things up, I love seeing kids get hit by animals. So a reward for being to have to put up with me is we're going to watch bulldogs kind of destroy little children. Okay, so the title of the talk are some of the challenges that we have trying to overcome neurological trauma and neurological diseases. So there are a few of them, and over standard lecture materials, we'll have headlines like first challenge, whatever. So the first major one is you cannot be a humanities major. So where do you think Spanish lies? In the humanities, right? So as a young 18 to 22-year-old idiot, I had no idea what I wanted to do. So I grew up in Columbus, Ohio, or just north of Columbus, Ohio. So everybody goes to Ohio State. You may not know what you want to do, but you get to go to 6 or 7 football games a year. If they beat Michigan, you get to riot and light a few cars on fire. So overall, a good college experience. So over time, you have to find out what you want to do. And Ohio State, being a 61,000 student, large land-grant university, has about anything you can want to do. So when you're 18 years old, you don't know what you want to do, you just take classes. And Ohio State at the time was on quarters. And even more so, if you took 12 credit hours, you were a full-time student, and let's say it cost $3,000 for that quarter. If you were a full-time student that took 24 credit hours, it cost $3,000. So there was no difference in taking all these extra credit hours. So I just took classes, hoping one thing would hit, and it never did. So I took classes in journalism thinking I could be the next Hunter Thompson. I took theater classes thinking I could be the next Samuel Beckett. Took literature classes and poetry classes thinking I could be like Dorothy Parker, right? I'm way too lame for all of that stuff. And one day you're looking in the mirror, you like have a beret in your hand, and like, am I a hipster? And it's kind of a depressing thing to think about. So I've always loved fitness. So I don't know if any of you saw me walking around, but I think one of the reasons I like neurological disorders is I have one. I had neuromuscular disease when I was a kid. I'm probably the only person, maybe in Florida, that is 40 years old or younger that had polio. So I was paralyzed for a few years, and then you make do. Luckily for me, it stopped right at my waist, so it never got up to the diaphragm. So I struggle to walk, don't really have any leg muscles. But throughout all of that, the goal is I want to be fit because two things happen with post-polio. With polio, you get post-polio, which you lose the muscles that you have, and you just become unfit. So I thought, well, I'm going to get as fit as I possibly can and enjoy the things that I want to do because, I can do them. But then the other thing is to build a reserve in case something bad happens, because exercise is really just essentially a bank account for your body. You don't really need to be some 240-pound super jacked dude bro, but having that muscle helps when you need it. You have a surgery, you're in a car wreck, you're on bed rest. Those things are important. So I think it was subconsciously, but exercise I've always done. Being physically fit makes perfect sense. So what if we can teach kids how to be fit? This was right at the beginning, so I'm not that old, around 2003 or 4, where the obesity epidemic is starting to go up. So how can you improve the lives of these young kids so they don't face the things that their older people are starting to face, becoming overweight, low levels of physical activity, and things like that? And physical education is pretty cool if you don't have a teacher that's just the football coach throwing stuff out. You learn how to work with teammates, you work on motor skills, you get to be fit. And it's as good as anything that you've ever had. As good as the teacher that you have is as good as you can learn. So I decided I'm going to be a PE teacher. That's a nice, normal thing to think. And then I decided that I didn't want to. And I went to Spain. So that's kind of where you get Spanish from. And believe it or not, that beautiful long blonde hair is my hair. That is me. I do not have that hair anymore. I had that hair for 5 years, and there is not a single day that goes by that I wish that I didn't have it. So that's pretty luxurious. And that was like a 4 out of 10 day. Like it was a showstopper. And I was in Granada, Spain. Has anybody ever been to Granada? So you have the Alhambra, you have the Albaicin, the most romantic place in the world. It doesn't rain. You can go ski in the mountains 40 minutes, and then you can go to the Mediterranean 40 minutes. It's about the best place in southern Spain. But what I did find there was a direction. Finally, after searching for 4 years of, am I going to do this? In a PE major, like the first person ever to double major in Spanish, linguistics, and physical education. Those two things don't usually intersect. But we had to take classes there. It was essentially high school, 8 to 12, then you go for siesta, then 3 to 8. A very long day, 5 days a week. And you pick up classes. I took Spanish literature, Spanish poetry, and I was exposed to this book called The Romancero Gitano. Whoops. So by Federico Garcia Lorca. So a well-known, these days he's more well-known as a playwright, but he's probably southern Spain's best piano player, believe it or not, and poet. So this is a collection of 18 romance-style poems about the Roma gypsy population around the rural part of southern Spain. Beautiful poems. I can still read them, believe it or not, even though I haven't taken Spanish in 20-some years. But within this kind of time course, there's this art history, this modernist art thing. This is where I've got the love of modern art. In southern Spain and the world, that was kind of combative to the Franco regime and this incoming encroach of fascism in Spain. And Federico Garcia Lorca was the metaphor for that. He was taken out into a field and shot in military style, as they did in the days. But the shooting didn't interest me. That kind of sounds weird. But the works of this group and his cohort of this artistic tide of Southern Spanish national identity, especially revolving around the Roma Gypsy culture, which is, Spain's identity is Roma Gypsy culture. It's flamenco. When you think of Spain, you think of flamenco. You might think of paella. That's probably logical too. But I think of flamenco. So that's where the love of Spanish history and why I decided I wanted to be a Spanish major. You could also take a full coursework of credit for a quarter and you get to live in Spain. It's a pretty good deal. So got back to Ohio State. The PE program said, you need to be here for a year and a half because you missed a class. And I said, well, that's dumb. I'm not going to do that. So I applied, graduated with my degree in Spanish, then applied to graduate school in this little program, the Iberian Literary and Cultural Studies program at Ohio State. I got in, but believe it or not, they did not tell me. I didn't know I got in. So I was super excited to continue this. I didn't hear anything. And it's like, well, okay, what am I going to do? I had you do what everybody does. You've finally got that direction that you want. I want to study this really cool Southern Spanish, very art niche, which is the most, artistic hipster thing you can do. And then you go to the University of Kansas and get a degree in exercise physiology. Yeah, so I did that. And as things go, you get weirdly lucky. I met a friend in Spain. She went to KU. She was a med student, or going to be pre-med student going to med school. Her campus job was to swipe the cards for people to get into the employee gym, which is in where the exercise physiology department was. She knew the faculty members, and I just one day just searched, I wonder what this department is like. Exercise physiology sounds cool. I don't know what physiology is because I'm an art nerd, but I know what exercise means. So you look into it, and I found out that if you get into their master's program, it's in the School of Education, so you become licensed to teach. So Ohio State said, you can't be a phys ed teacher. And so I went there and it's like, well, now I can be a phys ed teacher. And I get to hang out with my friends that I met when I was in Spain. Cool. Applied to that. Didn't hear anything. It's like, okay, I don't know if I'm going to go to grad school. Do I have to get a job? What do I do? So I call the program director. His name is Phil Gallagher. He turned out to be my master's and PhD mentor. He answers the phone. What? Like, oh, well, this is not a good start. Hey, I applied to your program. I haven't heard anything back. Are you sure you want to come? Yeah? All right, you're in. That was it. To this day, I have no idea how he let me into that program. If you would have looked at my transcript, which he clearly did not, I had 3 science classes, Bio 100, Anatomy and Physiology, Chemistry 100. That was it. That was my science background. It's actually quite astonishing that he has a leadership position anywhere. But regardless, I get into the lab and, oh, what's this? I don't know what this is. What's this mask for? And then within a month, working on a project, looking at muscle physiology of rats going downhill. Get my first author paper during my master's program. So I think technically I'm a scientist. It's like, oh, that's pretty cool. So right after this paper was published, 2 years after I started the program, get a phone call. 740, that's the Ohio zip code, or 614, Columbus, Ohio zip code. Because in 2009, you still answered your phone when weird numbers call you. Hey, congratulations on getting into the Ohio State Spanish program. So I was like, well, I applied a long time ago. Well, we got in, we thought you deferred. It's like, no, nobody told me. I'd actually gotten in a month after I applied. They just didn't tell me. So 2 years later, they're probably going through some weird log system. It's like, who is this guy and why? And they went through, no one ever told me. So that's how I ended up at KU. Pretty fun, because I would not have went if I would have accepted to the Spanish program. And then this kind of transitions in, okay, now I kind of have a line of research. Muscle physiology, exercise physiology. All right, this is cool. Now this is solidified. Now I'm kind of a real scientist. I have a PhD, 3 letters that kind of are officially you're a scientist. But KU was a very small lab in a school of education in a state school, not very successful. what I thought was science was not science. This was good work, but very low impact, very low throughput. So you go to the best place in the country to do clinical spinal cord injury work. And again, this is all luck and networking. And when someone, pretty much a kind of rule to just be a good person to work with and not be a jerk, because then people will come up to you out of nowhere and say, hey, I heard you were looking for this. I know someone. That's how I got the job here. So I had a disgruntled, weird person answer the phone and let me into a PhD program. And then this person took me off without any hesitation because I got good word from my department chair. So going to the Bronx was amazing. I got to learn how to be what is called, the tried and true scientist, writing grant applications, writing papers, mentoring students, but also having the greatest mentor I've ever had, Chris Cardozo, who was basically just like, yeah, do what you want, but we'll kind of control you when we need to. So you have a lot of independence, which is what you need, and more so, no fear of making a mistake, because that's a lot of scientists have. They're really, especially young scientists, you don't want to make a mistake. So luckily, he let me make tons of mistakes. I made a $15,000 mistake, and I went in and told him, and he goes, will you do it again? I don't think so. Okay, then you learned a lesson. Paid a lot of money for that lesson, but it was a lesson learned. So when that lesson is, when you see a step that goes A, B, C, and D, D's pretty important. And so now I start to get into pretty cool work. We're doing applied work, putting drugs into a mouse to try to preserve muscle health, doing muscle health in people with spinal cord injury with testosterone, then slowly getting into more mechanistic and cool things. So going from someone that had never held a pipette to doing high resolution molecular screenings of things within a mouse muscle within 4 years is pretty cool. So as you can see, all of this is spinal cord injury. I've spent 10 years of my early career scientist life as a spinal cord injury investigator. Almost all my publications are that way because that's kind of how I was mentored. And then ultimately, you get a big grant, big is relative. You get a grant that is big for you, and that's what you do. So I was fortunate enough to get funded through the VA to continue this work. And then a little brief break. And so you'll notice it is another bulldog. Yep, there we go. And it's just a dog being a dog. All right, now that's enough about me. Now I'm here, that was my life in 15 to 20 minutes. Now we're going to get into some data. So, whole career as a scientist and an art hipster, spinal cord injury. About 30,000 people in the US, or 300,000 people in the US have a spinal cord injury. It is not a rare thing. There are 18,000 new injuries every single year. So these injury rates aren't going down, they're not going up. 18,000 a year that kind of refresh themselves over time. So it is still a continual national problem. 85% of these are from three things: vehicular accidents, falls, acts of violence. Acts of violence can be a gunshot, it could be a stab wound, things that can penetrate the spinal cord to cause some damage. But by far, vehicular accidents, no matter how safe cars are, physics is pretty strong. Your neck is very weak. That's kind of how that happens. But an interesting thing is, as cars are getting safer, falls now are becoming more of a problem. Falls leading to spinal cord injury. And you can also kind of see this reflected in the age. Oh, well, it's a mouse. You probably can't read it. But the average age has gone from around 28 years to now 43 years old. All right, well, that was so shocking, like the computer had to restart. So the problems exist, and the problems are actually burdensome in a lot of ways. A lot of people have spinal cord injury. A lot of people are getting spinal cord injuries, and the age that people get their spinal cord injury is increasing. Let's not make any mistake about it. Spinal cord injury is still what we call, and I don't say this as a joke, but the dumb young male syndrome. 25% of all spinal cord injuries are still young males, vehicular accidents, speeding, drunk. Sports does occur, but nowhere near the rates of that. So 25% of all spinal cord injuries are still people aged around 17 to 22, mostly male. 80% pale. But if you have a high-level injury, that first year after injury is about $1.4 million just in medical costs. As the injuries get a little bit lower and lower, those medical costs go down. But if you're a young individual, let's say you're 25, that's reported here from data from UAB, your lifetime medical costs are about $6 million. Every year after you have a spinal cord injury, it's about $250,000 if you have these high-level injuries. So it is a sustained medical burden that does not leave. Spinal cord injury is also advanced aging. Their lifespan is reduced by about 10 years. There's a lot of things that go into that, the injury itself, the response to the trauma itself, a lot of those things. But a key one is that they're the most inactive population outside of people prescribed to actual full-time bed rest. Wheelchairs are very efficient. They're meant to be. They're wheels. There's no friction. It's pretty easy to wheel a chair. Let's say that you're in New York. There is a heavy snow. Can you get out of your house? Can you get to a car? Do you own a car? Can you get to a subway? You're restricted by your environment and your infrastructure. A place like Ocala or Pensacola you have your own parking lot, you may have your own car. The car has to be adapted to you. There's a lot of things that lead to people being inactive, but they respond very, very, very well to exercise. And probably the biggest part is that they just don't get better. You get your injury, and that's pretty much your injury. Very few people will improve from year 1 to 5, and very few people will improve from year 5 to beyond. You do recover from the time you hit the emergency department to about the time you get out of rehab. You can improve as the swelling around the spinal cord decreases and function comes back, but that 1-year time point, you're pretty much locked in. And this is why. The physical trauma to the spinal cord leaves what we call a lesion or a cyst. There's kind of a bunch of different ways to call it. I call it a lesion of scar tissue that neurons cannot transverse through. Theoretically, they can. In mouse models, it is not detrimental. So it is not a weird force field. You can get things through it, but it's very, very difficult. But in humans, it's just not that possible. So you have this gray fibrotic tissue that's non-neurons, but the body is trying to do as best as it can. It knows something weird's going on, and it's trying to clear it and make it better. It just happens to do what the body does. It makes a scar that cannot be repaired. And more so, the areas above and below that still have functioning neurons are now stressed. They're putting off inflammatory markers, reactive oxygen species, things like that are they kind of inhibit how these things grow. So that's what we're trying to battle against, this big scar that won't leave. There's 3 things that have been shown, and I just picked 3 that I thought were good examples. There are other ones. I think these are pretty cool. So we've all heard about stem cells and using stem cells to kind of regenerate this. The early stem cell therapies were not very good because you were taking donor cells, putting them in and having weird inflammatory reactions and growing weird lumps that actually sometimes made it worse on the spinal cord. But these individuals, which was a study done recently, as soon as they were, unfortunately, spinal cord injury is a spontaneous thing. Nobody's walking down the street with a sign saying, "Someone please give me a spinal cord injury today." So you're in the ED or the ICU, a researcher comes in, they get you to sign an informed consent because you're now a research participant. They take your blood, they isolate some stem cells, and then while you're in your surgery to stabilize your spine, they're putting your own stem cells back into you, hoping that it will improve your function. So in this study, it was 13 people that they were able to go through. And these marks here, kind of like a stoplight. Red is bad, orange is and green is good to go. So over time, you can see that these 13 people, these 2 I just cherry picked, were able to improve their sensory function. So these colors are the AIS scoring. So you take a little pen, and if you can feel them, they're in these little ranges. But for some people, it doesn't work. These are humans. Like, humans don't respond to things exactly. So I consider this kind of biotechnology, using molecular biology, cell biology to try to help yourself. But you just have to be, unfortunately, lucky enough to be in a hospital that's doing this study, and they take your own blood, all that stuff. What I think is the coolest, you may have seen this one because this was in all the major news stories last summer. So there's this group out of France that does gnarly stuff. So this is led by Jocelyn Bloch. She's a French neuroscientist, neurosurgeon. And this individual, so they've only done this once, they basically made a digital spinal cord. So how do you do this? Well, some person that wants to have implants put into their brain says, please put these 2 implants into my brain. So that's the top part. Then what they do is over the course of, I think it was 4 months, it may have been longer or shorter, I think it was 4 months, they ask questions. Think about walking. Think about walking. Now actually try to walk. And a bunch of other questions, and they use very sophisticated computer science to basically map how a brain makes a decision. And then to understand that this electrical signal equals walking, this electrical signal equals thinking about walking, and so on. Because the last thing that you want is someone to think about walking and then their legs start moving. So it's a very weird philosophical, cool question that they were able to resolve. So the bottom part, surgery number 2, they put a custom-made stimulator onto his spinal cord. And within those array patterns, you can hit, sorry, I talk with my hands even though I'm talking to people. There's a pattern that your spinal cord uses to walk. And they mapped his perfectly. So basically now he can think about walking. It sends a signal from that. This is Bluetooth, like, right? He's walking with Bluetooth. It sends a signal, gets processed by that huge laptop that's at his backpack, and it tells the spinal cord to walk. And on the right panel, the blurry picture is when they turn it off. So this person basically started functionally ambulating again using wireless technology. Like the same technology that allows you to like control your microwave with your phone. Yeah, that's what's allowing him to walk. There are major limitations to this though. It was one person that had motor incomplete spinal cord injury. So he still had movement of his legs, he still sensed things. This is probably still not possible for somebody that has a very severe spinal cord injury, but I hope that they prove me wrong. The third, I'll cover the, okay. Anybody in here a lamprey or know what a lamprey is? So it's a weird, oh, well, I guess it's right there. So it's a very primordial looking animal that to my knowledge, and my knowledge of Spanish literature is really helpful in these cases, the only animal that can actually fully functionally regenerate a spinal cord if you cut it. So someday somebody said, let's cut into a lamprey, and did a study to find that that's what happens. The lamprey, you can take a pair of surgical scissors, sever the spinal cord, and then 9 weeks later, perfectly all functional. With the histology, you can kind of see the greens are the axon. So morphologically, it's not perfect. There's still some gaps, there's still things around, but functionally, it's back to normal. This group then took the same lamprey, cut open the spinal cord at the same site, cut it again, and it still regenerated. So the lamprey is weird. I mean, it does share 3 genes. It has 3 genes that are conserved in humans. So the spinal cord that we have has 3 things that they do, but probably not the best model system. Oh, of course. I'll leave you in a little bit of suspense. So this wood here was done by a collaborator at Feinstein Medical School, which is the medical school of Hofstra out at Long Island. I wasn't a part of the study, this is just a collaborator. So this is a lamprey that had a spinal cord transection, so paralyzed. This is 9 weeks later. So 9 weeks is pretty fast. For a lamprey, it's a long time, but so can't tell. So lamprey, pretty cool. I don't know if I want to be a lamprey neurologist. And then we'll get to the fun one. To be fair, this is not a bulldog. So we've been talking about the spinal cord itself. We are way more than the spinal cord. So Bill Bauman and Ann Spungen, who are here, led the center that I was in. They were a VA-funded center for 22 years, incredibly long, the center of excellence that I worked in. These are the things that they found were dysfunctional in people with spinal cord injury. So they were pioneers of understanding the unique things people with spinal cord injury face after the injury that aren't directly related to the injury per se. It's not the central nervous system. Of course, it's related to it because these tissues and organ systems no longer have innervation. but they've developed treatments for all of this. So these are all the things that someone with spinal cord injury likely has to deal with on a day-to-day basis. So I'm a muscle physiologist. This paper came out last year. And this just shows what happens if you just basically make the adrenal gland silent. So you do an injury above the adrenal gland and you do an adrenal gland below it. 3 vertebrae in a mouse. That's it. Or sorry, 6 vertebrae. I can count from 2 to 9, from 9 to 3, even though I'm a Spanish major. These are functionally and wholly different injuries. Thoracic spinal cord injury, same cut, same scissors, same injury model, 6 vertebrae different, and it's essentially a different disease. And you can look at this by body weight, which is at the top. So the mice with the T9 injury, so that low back injury, actually kind of recovered pretty well. The others didn't. If we look at this molecularly, if you just look at the numbers, you're like, yeah, that's a big difference. But they don't actually lose the nerves that connect their muscle. They stay intact. They just are not active. So that's why someone can have an epidural stimulator put on their spinal cord and walk again, because they don't lose nerves. But these injuries, so which model do you pick? Do you pick a lamprey to learn something? These are in mice. Mice aren't humans. Rats aren't humans. Do you use something that's like a human? Well, if you want to work with a pig who has a spinal cord similar to a human, you have to have a team that can lift a 250-pound pig. You have to have an area for the pig to walk. Or what about non-human primates? So this model system has become very complex because the injury itself is very complex. Then, it wouldn't be IHMC without showing myself in a robot. So this is me. So people with spinal cord injury can utilize can use an exoskeleton in the house. There are 2 FDA-approved exoskeletons that you can use for community and home use. They are very, very, very restrictive. And I'll show you data that came out on Thursday of why it's such a problem, why we don't see paralyzed people with exoskeletons. The primary one is, you can see right here, there's a team of 4 around me. 3 of them are collecting data and 1 is being safety. But I also have crutches. The FDA mandates safety crutches. You cannot use an exoskeleton with spinal cord injury without crutches. You can use an exoskeleton to stand, but you cannot walk. Or can you? Oh, thank gosh. The music behind this is horrible, so I'm glad that it's quiet. So this is a company up in Toronto. The music might make it a little bit better. So it's a self-balancing exoskeleton. No crutches. This is not FDA approved, so keep that in mind. It is massive and bulky, but the importance of exoskeleton uptake is because you want to use it in your house. You're cooking, you want to reach into a cupboard, you want to reach into a fridge, something like that. So if you talk to people with spinal cord injury, the wheelchair is trusting. They like the wheelchair, they know what they can get out of it. But the burden of using an exoskeleton in normal everyday home use is just really not sustainable. And this is published, and as well as anecdotal. So as I said, new data came out on Thursday by my old colleagues up at the Bronx. They did a huge study where any veteran with a spinal cord injury that wanted an exoskeleton and qualified got one for free, an $80,000 device for free. Now you would think, why would the VA give $80,000 devices away? Well, if you can remember, that is chump change compared to the annual medical costs of people with spinal cord injury. That $80,000 could potentially save money. So what happens if you give someone an exoskeleton and put it in their house and say, here you go? Nothing. Nothing. They actually, in some things, did worse than people that got nothing. So you would ask, okay, now they can get up, they can ambulate, they can walk, they can exercise, they can do all this stuff. And I will tell you, they average 1.5 miles per month of walking. And the next sentence says why. You have to have a companion. If you do not have a stable life partner, if you don't have a a friend, a son, daughter, someone that can be around you, you're not going to utilize this thing. And more so, you cannot use it. So what is the point of a device if it's not useful? So this is the main reason why you don't see people with spinal cord injury up and down the main drags. There are technological barriers that just cannot be overcome. However, just getting upright in an exoskeleton, even if you walk that 1.5 miles per hour, these people had better feelings of quality of life and were more comfortable with their injuries. So there is substantial value in getting an individual upright. A lot of it's psychological. Every day, all day, they're looked down upon. You may not think of that, but if you've taken any art class, like I've taken 5 billion of them, it is a common cinematic technique for someone in power to look down. So if you've ever noticed a weird tension in a weird like Scorsese film or something where the camera shifts, it's a psychological trick that people with spinal cord injury face every day. So just getting upright was beneficial, even if they only walk 1.5 miles per month. All right, I don't know if I have another animal thing. So this will be a surprise for everybody. My whole career has been mice. 10 years of just working in vivariums. So it's pretty fun, especially when you get a successful grant and you no longer have to touch them. So that's always kind of fun. You pay somebody else to do it. So join IHMC. IHMC, believe it or not, is not well known for their animal work. We don't have a vivarium. UWF doesn't have a vivarium. I still do animal work, but my collaborators handle the animals, and I get to work with the tissues as I want. Pensacola is not UAB, or it's not any city that has a major biomedical infrastructure. There's no world-renowned medical school. That's what UAB was. That's what UF is. I could go to UF and do all of this stuff, and I have many collaborators at UF doing spinal cord injury work. So, not there. So I've spent my whole career as an art moron, preclinical mouse biologist, and now I'm coming to a place that doesn't offer any of those. So what do? Well, you look at the state of Florida and you go, neurological disorders, and I don't mean this mean, are kind of cool. Having them is not cool. Studying them is pretty cool. Well, Florida has the highest rate of Parkinson's than any place. Number 2 in total population, because California has like 700 billion people. But if you look at the percentage of the population that have Parkinson's, Florida's number 1. Whoops. And more so, where Pensacola is, around Escambia and Santa Rosa County, has high rates of Parkinson's, even for Florida. So Parkinson's is, it's a genetic disorder when you know what the gene is. The other things are genetic things that we don't know of that interact with your environment. So something weird is happening in Pensacola, in the surrounding areas. So it's like, well, you filter your water and you make sure you buy your vegetables from somebody else. But the data are there. There's high rates of Parkinson's. So I'm going to try to study Parkinson's. That's like a sane thing to do. So when I was recruited down to work at IHMC by Marcus Baumann, one of the reasons that I joined his lab at UAB was to do exercise studies. I wanted to be in spinal cord injury because Marcus led the UAB Center for Exercise Medicine. And then he recruited me to Birmingham, super excited to work in his lab. 3 months later, he retired. It's like, oh, well, you bastard. And so now I'm homeless because the grant that I had to have a lab in. Marcus just retired and moved to Pensacola. But then he showed me what he was working on. And it's like, okay, I think I can do this. And Marcus was the bridge to Parkinson's work because he has done some innovative work with resistance training in people with Parkinson's. So just as a quick tutorial of Parkinson's, it does not start, it is clinically diagnosed when you have tremors. So the actual diagnosis is this rigidity, tremor, and bradykinesia, so slow walking. But you get Parkinson's way before. It starts in the gut, and it starts with sleep dysfunction. So it is Parkinson's in this prodromal state can be undiagnosed other than if you go, I haven't been sleeping well lately over time, and then my gut kind of feels weird. As that goes up, you can see That's when things like depression, weird problems with drinking water, dysphagia come in. And then it goes into what we think of traditional Parkinson's. You get your motor defects, usually start in one limb, then they go to the other. Then you start to have cognitive problems, and then you start to get into the very severe Parkinsonism and death. So our goal is to use exercise to, you're not going to slow it down. Like you might be able to increase the health span, you're not stopping Parkinson's. But how can we extend this healthy window where they're independently functioning to improve their lifespan and quality of life? So target the mitochondria. So if you can remember from Biology 101, the one thing that I remember from undergrad, the powerhouse of the cell, right? Mitochondria are the powerhouse of the cell. So the genes that we know cause Parkinson's affect the mitochondria. They prevent the ability of the mitochondria, which is the thing that makes most of the energy you use on a daily basis. It prevents the turnover of it. So you have this very weird kind of subcellular glom of just bad things. That's the scientific way to put it, bad things. So these are the two things. I'm pretty sure you didn't think you were going to get a molecular biology course when you came today, but Parkin, the Park gene, is one of those. It's the thing that regulates how mitochondria turn over. And so this is all that this is showing. You have rapid, you stop the removal of poor mitochondria, essentially. But it goes beyond that. Even when you don't know the genetic cause of Parkinson's, if you affect the mitochondria, and this is a mouse model, or will be a mouse model. So if you can't remember how the mitochondria work, all that you have to know is you get ATP. There's 5 complexes. This is them. Predominantly, all of the food that you eat is meant to drive this process. So if you inhibit the green one with a drug, or in a mouse, you knock that gene out, or this thing called complex 1 has 45 proteins in it. If you knock one of those proteins out, you get Parkinson's. So that's all that this graph is showing. So if we look at the fancy green little maps, they're tracing how a mouse walks in a cage. So mice do not walk as they get further and further progressed. If we look at the top right, a very easy thing you can do to a mouse is you put a sticky pad on their paw. A mouse with a lot of motor control can just grab it, take it off, or they can chew it and take it off. But as the disease progresses, they lose that fine motor skill and actually can't remove it. And then the other things are similarly showing. So basically what this study said is we know that the genetic cause of Parkinson's is mitochondria related. And they showed a theoretical thing that if you just stop the mitochondria from functioning in general, you get Parkinson's. It's a mouse. This is not Parkinson's. It is a mouse model of Parkinson's. Because 95% of what we know as Parkinson's is called idiopathic Parkinson's. It doesn't really have a known genetic cause. There are certainly genes involved in that. We just don't know what they are. But the tried and true thing that if you go to the doctor and they said, you have a mutation in this gene, you have Parkinson's, is a very, very small amount. And if you do a Google search, so I just literally stole this off the internet, of what people think a mitochondria is, it's this little bean thing. Well, do we think that it is? No. I'm interested in muscle mitochondria. The mitochondria make up about 15% of your muscle. Your muscle uses a lot of energy. For your heart, it's 50%. So because your heart, well, hopefully all of you want your heart to work. You need a lot of energy. So most of your heart is mitochondria. A lot of your muscle is mitochondria. So everybody just thinks mitochondria is this little bean thing that sits in the cell and it makes a lot of energy. But if we look at the muscle, well, this isn't some weird seeing eye test. So if you look at the muscle, this is actually what your mitochondria is. It's a very dense connected network of a large singular connected tunnel, essentially. When it starts to get diseased, it gets clamped off and recycled, and then it reforms again. So it's not one little isolated fragment. It is your whole tissue connected. That's all that this is meant to show. I think it's really pretty, but basically saying that mitochondria are very complex. That's why it's so hard to target them. But also, if you have a defect in those or defect in how you handle those, you can see why it's such a problem. So this is Marcus's data. I'm not going to pretend that it's mine, but it has kind of spurred us. So we use we're exercise physiologists, and we are not gentle. I don't care what disease you have. Your body responds to intensity. We want you to be loaded, doing maximal contractions. We want you to run as fast as you can, to lift as much as you can. We do not believe in just kind of guiding somebody along. So even in people with Parkinson's, we crush them. We want them to do some work. And so this is Marcus's first study that they did, high intensity exercise with people with Parkinson's. This is resistance exercise, getting them under a weight, getting after it. And the left are all the clinical scores. So their clinical scores improved over 6 months or over 4 months of training. So the symptomology of Parkinson's was reduced after this exercise training. The middle panel there are their mitochondria function. So you can see it's below the normal average. So that's what that grayed out box is. We actually improved their mitochondrial function. And on the right, we improved blood flow. I say we, Marcus, I wasn't even in the state at the time. Improved the blood flow to the parts of the brain affected by Parkinson's the most, the prefrontal cortex and the substantia nigra. So exercise is incredibly important for those with Parkinson's. This study has been repeated. We're working on the manuscript right now. We show both the physiological changes, so they respond well, they get stronger, they walk faster, they have more endurance, but also ancillary things. They have less feelings of anxiousness, they're less depressed, their quality of life is better, their sleep improves, the density of how well they sleep improves. So this isn't just about getting your muscles stronger. Exercise, especially the type of exercise that we ask these participants to do, is a whole body, holistic approach to improving the health of an individual and improving that little health quality and trying to extend their health span. So I think that's it. So main summary is I hope you're a lamprey. I think you never want to be in a situation where you're like, well, today I wish I was a lamprey. That's probably not great. But for spinal cord injury, there are numerous barriers to trying to actually overcome that traumatic event. So the goal is to use things like exercise, technology to bridge that lesion, both technologically, biologically, whatever, to get function going, because that's ultimately what we want. We want people to have healthy quality of lives. We don't want a disease to restrict them to enjoying the life that they have. And Parkinson's, mitochondrial disease, we think about it as the brain. And that makes sense. That's kind of where all of the major things happen, as you get dementia, things along those lines. But it's a mitochondrial disease. Mitochondria dysfunction in every cell type that's ever been investigated in Parkinson's. Blood cells, bone cells, fat cells, all of them have mitochondrial dysfunction. We don't want that. You want healthily functioning mitochondria. And again, exercise. Please. So these are the acknowledgements. This is a small group of people. Obviously, I've been helped multiple times along the way. Most of the work that I presented, that was myself, was funded by the VA, those 2 grants. But yeah, there we go. Thank you very much.
I think we probably, have some questions.
Was that 50 minutes on the dot?
Okay, I've got you and you and you. And 1, 2, 3. Ready?
Thank you, Lori. Dr. Graham, did you begin your studies of the spinal cord injuries in any way because of your polio? And if you did you learn anything that helps you manage your polio?
No, I think it was, one, dumb luck of not getting accepted into some weird Spanish degree. I honestly think that's what it was. And the love of exercise is what drives that. So the polio virus is a motor neuron disintegrator. So those motor neurons aren't coming back. So this is as functioning as I'm going to be forever. So there's really no way to get around that. My guess is probably when you're like, you see somebody struggling to walk, you probably have a little empathy with them, like, oh, I kind of get that. Or in New York, you have so when I lived in Astoria, Queens, one of the most famous parts of New York, there were no elevators on the subway. You could literally not get from Astoria to Manhattan if you were in a wheelchair. And a part of that is like, I'm lucky I can go upstairs, but it's like, get a little pissed. Like you see someone in a wheelchair going around, or not even a wheelchair, someone with a new kid in a crib. So you kind of empathize a little with people, but luckily I can climb stairs. I can fall downstairs as good as anybody. So there are things that I'm cognizant of, but I think the interest in it is reinforced by that. But I think the thing that, as a scientist, that I'm very fortunate is not many people are paid to be curious. I find a lot of things interesting, as you can see from my background. So you see something new in front of you and you have the opportunity, if you're curious enough, you can find something to do with it. So probably I would say in the back of my mind, it's like, yeah, you're doing this for that. But it was never intentional. It was just some hungover professor at KU that was like, all right, come here.
Catch?
Okay. Dr. Graham, are there any commonly used prescription drugs that could inhibit the mitochondrial function and therefore down the road lead to Parkinson's disease?
So we have the answer to that is no. There was a drug that we have tried to get a hold of called elamoprotide. It is a mitochondrial targeting little peptide. It was actually aim one of my VA grant was to use it with spinal cord injury. It did not hit their FDA, a scientist and clinical threshold. It didn't reach that P05 that it was effective. So theoretically, molecules like these exist. They are not FDA approved. There is an FDA-approved nutraceutical called Urolithin A. It's a food supplement. It's made by your gut microbiome. It stimulates that process of mitochondrial turnover. We are very interested in that, and we hope that one day we can put it towards a Parkinson's study.
Are there any particular exercises that we can do at home that you highly recommend? And I speak about at home, something simple, setting, no machinery that I can do. I wake up, I do.
So one, it has to be fun. So one of the things that we combat is you try to tell people you have to lift 3 by 8, and you have to do this, you have to do deep squats, and you do heavy squats, and you do it once, and you don't do it again because it's not fun. I mean, it's the humans don't like it. They don't like to do that. That's perfectly fine. There's freaks like me that are like, oh yeah, this hurts so bad I can't walk, and I can't wait to do it again tomorrow. But that's a rare portion. Exercise adherence is a psychological problem. It's just not the U.S. It's everywhere. We call it the January 15th syndrome, right? Everybody can work out for 2 weeks. There is a legitimate scientific, 2-week thing that humans struggle with. How can you not develop a habit? But to answer your question, what is most fun? That's up to you. Exercise is different for people. For some people, walking is not exercise. For some people, walking is substantial exercise. So at home, you have a laundry basket, squat down, pick it up. That's a deadlift. You don't need some 400-pound bar or stuff like that. A book's heavy, like, oh, I'll lift this book. The goal is to be active. We talk about exercise because exercise is planned, prescribed, you do it, it's a routine. But really it's physical activity that we try to get people to do. We don't want people sitting. Believe it or not, my goal here, instead of doing those bulldogs crushing children, was to get you up and to do stuff. But I thought that might be a bit embarrassing for y'all. Because if that's, you don't want to be sitting down for an hour. I don't sit down for 10 minutes at work. Anything you do is good. You can be creative. This basket's heavy. 2 weeks later, oh, this basket's lighter. I'm going to put another basket in it. Things like that.
Your talk.
Correct. Yep.
Did you have enough? Yes, speak into it. Okay. Did you have enough subjects to do any differentiation of their ability to use their
mitochondria efficiently. As you know, related to IHMC, there's a tremendous amount of nutritional information now that suggests ways that people can dramatically increase the amount of ADP that their mitochondria are making, specifically by reducing the carbohydrate intake that they have. Did you have enough subjects to be able to look at that at all?
So this study was done the one that I'm presented was done from 2011 to 2014. So I would have been writing my PhD, dissertation. The one that was repeated at UAB. So full disclosure, I am taking advantage of a lot of work that other people did. You get lucky and you go into a lab and they say, hey, we just finished the study, we need it written up. Will you do it? Like, will I be first author? Yeah, then I'll do it. So you take a lot of work for that. That's not true though. We've done a lot of work. Nutritional interventions are always good, but very similar to exercise. Humans do not like, Americans do not like low carbohydrate diets. There is efficacy for ketogenic studies for things like that, but they still pale in comparison to what exercise can do. Those free fatty, or doing keto diets, low carbohydrate diets are certainly beneficial in the short term to kind of get your mitochondria started. But if your mitochondria are dysfunctional through some genetic problem, you need something more than just the ketogenic diet. Exercise is the best for that until they find a drug that is a mitophagy full-on activator, in my opinion. Ken would disagree.
I just wondered if you had done any work or heard about any work with essential tremor, and is that in any way related to the symptoms of Parkinson's? And I've heard that the ketogenic diet has some effect on that.
It may. I don't know anything about it, unfortunately.
All right, we see a lot of hands. We're going to do 1, 2, and we had a hand up over here. So we'll have to.
Oops, sorry. Dr. Graham, I noticed earlier in one of your slides.
I can't hear you,
Oh,
sorry.
I'm sorry. Yes, I noticed in one of your slides, your earlier slides, you had some information or a statement about a molecule known as Boldine. Mm-hmm. So I'm wondering, how does that play into your research or your topic?
So full disclosure, this is what we've patented. So this is the Boldine treatment. So I won't go into it too much. I'll talk to you afterwards if you'd like. But we have found that this is a phytochemical that's found in the Chilean boldo tree. As things go, indigenous people have been using this thing for a while in Chile. And we have found for some reason, well, I know the reason, I don't want to get into it, that when you administer this to an animal with spinal cord injury, they recover locomotor function. So I can talk to you later about why we think that's happening. And we've done some transgenic models to show the mechanism. So that was the data that I haven't presented. But it reduces pain, but it's a systemic oral drug. It is unbelievably insoluble in water, so it will never be in a human. So that's one of the other difficulties of translating these things. You find something really cool, it's really efficacious, and then you just go, this will never work in a human. Cool science, maybe you can iterate it with medicinal chemistry, but that's one of the limitations. But I'm happy to talk about it more later.
Hi. Thank you for your humor. It helped keep our attention.
Yeah.
And I have a two-part question. Could you speak for a minute about the connection between the loss of sense of smell and Parkinson's as a symptom maybe, or is that, you talk about it. And are you familiar with the PPMI study through the Michael J. Fox Foundation?
Yep. So the gradation of symptomology of PD are, things just like you said, as the nervous system starts to become dysfunctional, as these bodies start to progress up through, loss of taste, loss of smell are things that happen. So that's just coupled with disease progression. The PPMI is amazing. We've tried to leverage them to get data. They do high quality molecular work that tries to track the progress of Parkinson's disease. So we are very interested in it, and we would like to leverage, well, we have leveraged that data before. Oh. Yeah.
We would like to thank Dr. Graham for being here.